Hello, everybody. Welcome to lecture 17, I believe, right? Yep. Talking about glycolysis. And this is a pretty long one, but I feel like there's a lot of stuff in here that's not super necessary. Like, this is just intimidating to look at when you first see it. So we're kind of just going to skip over it and we're going to move on to stuff that actually matters. Um, again, kind of unnecessary. I get good to know that you need glycolysis for human health, and like a lot of deficiencies in these enzymes cause disease, but nothing really that important. You got different different fates of pyruvate. So you got glycolysis first into anaerobic fates of pyruvate, and then catabolism of fructose, and you kinds of stuff. So fructose is a bit different from glucose, but yeah. Yeah, so glycolysis, occurs in most organisms. I don't want to say all organisms because, of course, there's exceptions in biology, but um, used by both aerobic and anaerobic organisms, and it turns one molecule of glucose into two molecules of pyruvate because it's 6 carbons, right? Two molecules of. They've done a weird thing with the parentheses here. Formatting of these slides is always weird. Occurs in the cytoplasm. That's the other thing. You'll learn about citric acid cycle later, which occurs inside of the mitochondria, and you can shuttle things in and out of the mitochondria to help with these processes. And 10 enzymes catalyze the reaction. Go over those later. So, glycolysis, right, uses glucose. You can obviously use other sugars, and you can kind of stick them in as intermediates. For example, one of the intermediates in glycolysis is fructose 6 phosphate. So you could theoretically take a fructose, right? You could add a phosphate to it, fructose 1 phosphate, for example, and isomerize it to fructose 6 phosphate and go through glycolysis like that. So this is not just glucose, but glucose is the one that we think about when we hear glycolysis. So you got all sorts of things that can make glucose. Lactose, right, makes glucose into galactose. Remember, galactose is just an epimer of glucose, right? There's just one carbon difference. Sucrose, right? You got fructose and glucose. You should actually have these structures memorized, in my opinion, or at least try and memorize the Fisher projections of them. Makes it a little bit easier to figure out which ones are which and what you're looking at. You got starch and you got glycogen. We don't have starch because we're humans and we're not plants. Basically, we actually cannot digest. Can actually digest that, but anyways, yeah. So these got alpha 1,6 glycogen. This is what we do use as our storage system for sugars. So this is what we use. We got alpha 1,4 and alpha 1,6s. But yeah. Right. So here's some things to know. I would say if you were to know, I feel like a couple of so GLUT2 is for sure quite important, and GLUT4 are the main ones that you definitely should know. GLUT3 is helpful, and GLUT1 is also kind of helpful as well, but I would say GLUT2 and GLUT4 for sure. So GLUT2 liver, GLUT4 muscle, GLUT3 is brain, very high affinity for glucose. But yeah, you should know that GLUT2 is in like liver cells, right? It's got very low affinity, which makes sense because the liver is a storage system for glucose, so you don't want to actually store glucose if you don't have a lot of it. Very different from the muscle cells. If you got a little bit of glucose, then your muscle cells want to use up that glucose, right? So they have very high affinity glucose. So basically, right, if you have a ton of glucose, right, the GLUT2s will actually be able to take up the excess glucose and store it. And if you have very little glucose, your skeletal muscles can use that glucose, and your liver will just not bind it. The liver cells, particularly, will just not bind it, right? GLUT3, your brain uses a lot of ATP. If I'm not mistaken, I think it's 40 something percent of your ATP created in your body is actually used up by your brain, which is insane. Be wrong on that, but I vaguely remember this. And then GLUT1, just a general transporter, right? All over the place. Not quite as specific as GLUT2 and GLUT4 and GLUT3. So I would say no GLUT2 and GLUT4 for sure, and then keep GLUT3 in the back of your mind for sure, because that one definitely could show up. Like why does the brain have high affinity for glucose or something like that? What channel is responsible for glucose uptake in the brain, that kind of stuff. And then GLUT1 is pretty all-purpose. I feel like you probably won't need this one, but might be helpful to just know. Now you're getting to actual glycolysis. So There's really not much I can say here other than just memorize them. I can try and like explain some things, but yeah. So I like how they had, like, you can see the red line here because they didn't like properly. Anyways, so hexokinase, right? First up in glycolysis, it basically activates glucose and keeps it in the cell. So you can think about the type of you can think about what glucokinase is doing. So glucokinase is for the liver, but hexokinase, right? What it does is it takes glucose and turns it into glucose 6-phosphate, right? Turns it into this using ATP. And it uses magnesium as a cofactor, very high delta G in this direction, or very negative delta G in this direction, right? And why is this important? This is important because, you know, we've got a kind of an equilibrium. We've got glucose. If you got glucose, which is G outside the cell, right? G inside the cell, right? And the G inside the cell is actually being added to ATP, right, to make G6P, right? And so basically what's going on here is that as we make more G6P, right, the glucose inside the cell actually goes down, right, if that makes sense. And then what that results in is more glucose from outside the cell actually shifts this way, right? Basically it's an equilibrium, right? Because we are making G6P, right, you're actually decreasing the concentration of glucose inside the cell. Glucose, right, not glucose 6-phosphate, glucose. And because you're decreasing this concentration, there's a bigger driving force for glucose to come into the cell. Right? So it activates glucose and keeps it in the cell. Right? Once it's glucose 6-phosphate, it doesn't really leave. Right? And there's other types of hexokinase, right? There's type 1, type 2. They're all inhibited by G6P. Right? So these guys here are inhibited by G6P. But glucokinase, right, is low affinity for glucose. This is the liver one, right? Type 4 glucokinase, and is not inhibited by G6P. So you should be able to think about that in terms of feedback inhibition, right? So if we have a ton of glucose being turned into glucose 6-phosphate, we don't want to make more glucose. So we're just going to go back and inhibit this enzyme, right, and stop it from making more glucose 6-phosphate, right? That should make sense, right? Feedback inhibition, right? If we don't need something, we're going to stop making it. And the liver does not have that function for inhibition of that enzyme. So that's something to keep in mind. Definitely could ask you about the difference between upper muscle and liver cells. One of the main differences here is the difference in glucose 6-phosphate, or a difference in hexokinase's inhibition, and also the level of affinity for these different enzymes. So glucokinase has a much higher affinity, generally speaking, or much lower affinity, sorry. Glucokinase has a much lower affinity than hexokinase, and glucokinase is in the liver, and hexokinase is in the brain, skeletal muscle, and everywhere else, right? That should make sense intuitively, right? Because if the liver is a storage form, right, you don't want your storage form using up all your glucose to store it if your muscles need the glucose, right? So it makes sense for all of the liver enzymes to have a slightly lower affinity in the case of glucokinase, right? So that they don't just take every single piece of glucose and store it instead of actually using it for the body's processes, right? So hopefully that makes some sense. The other thing I want to bring your attention to is cofactor. So generally speaking, whenever you see ATP, magnesium is used as a cofactor, and the reason why is because ATP, right, adenosine triphosphate, those phosphates are negatively charged, right, and the magnesium 2+, the 2+ charge, right, basically establishes an ionic interaction with the ATP in the binding site, which allows for faster catalytic activity, right? Hopefully that makes sense, right? This is basically how all enzymes with ATP or GTP or any of those kinds of TPs work, or DPs, honestly. But obviously there are exceptions, so all of them, but it's generally a good rule of thumb. Yeah, so we refer to the investment phase and the payoff phase. So the investment phase, right, basically what we're doing is we are investing some ATP, right, and it's basically this first 4 or 5 steps of glycolysis, right? And the payoff phase is this back end stage here. So it's called the investment phase, right? We're investing 2 ATP, right? And the payoff phase, the main thing here is we're getting pyruvate, of course, right? And we're getting 4 ATP. Right? So we get a net 2 ATP. But the payoff phase, right, you're producing NADH. You're producing technically 4 ATP, right? But because this actually happens twice, so you have to think about it. So this is only showing that this glyceraldehyde goes through, right? This DHAP also gets turned into glyceraldehyde, right? And actually goes through again, right? So there's 2 glyceraldehydes that actually go through this process. So you actually get 4 of these, right? This is actually a typo. Actually, no, sorry. Hold on a minute. Oh no, sorry. They showed it down here. Never mind. I lied. Yeah, they showed both down here. So each one generates 1 ATP. So there's 2 ATP here and 2 ATP. Right. So 4 ATP total generated, right? And then yeah, that's the main thing there. You also generate these NADHs, these reducing agents, and then of course pyruvate, which goes on to the citric acid cycle, and pyruvate dehydrogenase. So second step is phosphoglucose isomerase or phosphohexose isomerase, whatever one you want to use. I personally learned as phosphohexose isomerase, but it does not make a difference. They're all the same thing. So all that we're doing now is we're isomerizing this glucose 6-phosphate into a fructose 6-phosphate because they are isomers of each other, right? To be relatively straightforward, right? There's not really much else there. Most of these carbons you can see actually stay the same, right? There's a couple changes, but for the most part, nothing really changes that much. Basically, right, this, I guess I'll say that, like, this bond right here basically moves and actually attaches here instead, basically. But you don't really have to know that, like, very minute structural, like, interaction. I would just say you should know, generally speaking, what the two. Right? And you should be able to, you can just draw glucose and draw fructose, then add the phosphate, and you should have an idea of what's going on, right? Like, draw out the Fisher projection, see how those are different, right? But, yeah. Basically, we're just going from the aldose form. There is to it. The other one, actually, one more thing I want to say about this step here is that there are 3 very large negative delta G steps, and this is one of them, the hexokinase step. And the reason being is that the hydrolysis of ATP generally, right, carries a lot of negative energy with it. It's like negative 30 kilojoules, right? So that's usually enough to overpower a lot of these non-spontaneous reactions. So the fact that ATP is being hydrolyzed here is what basically allows this reaction to proceed. It's a coupled reaction, right? Adding a phosphate group to this is not very favorable, but hydrolyzing ATP is. If you add those delta G's together, it's an overall negative value. So this step, pretty straightforward, I would say. You just isomerize the glucose 6-phosphate you just made into fructose 6-phosphate using phosphohexose isomerase or phosphoglucose isomerase. So yeah, he goes through all of this, right? I don't think you need to know this entire mechanism. What I would say that you should keep in mind is basically, right, we're starting with our glucose 6-phosphate. We're opening up this into our Fisher projection, right? And then we're basically doing an isomerization between this carbon and this carbon. You can see here, right? You're basically doing some form of enol reaction where, like, maybe this bond goes here, right? This bond goes here, and then this bond goes up, and you're basically forming a ketone, right? Do you need to know that whole mechanism? No. Again, I think it's just important to keep in mind that this is the reaction, and this is generally how it goes. But yeah. Okay. PFK1, phosphofructokinase 1, another ATP driven reaction. Most likely has a magnesium as well in this process, is not shown, but if they ask you about, oh, what cofactors are available in this process, you could definitely say magnesium as an option, I think. I think that would be accepted. I think it's totally valid. But yeah, let's go ahead. Let me double check that because I don't want to make sure I'm not lying here. Yeah, it does. Yeah, generally speaking, whenever you see something that has ATP on it, it usually requires magnesium or GTP or TTP or any of those triphosphates, right? So just something to keep in mind. Anyways, so moving on, right? So fructose 6-phosphate, another just kinase reaction, right? Nothing crazy here. Just adding a phosphate group onto this side. Now we had one over here already, so we just add one to this side as well. Yeah, this is fructose 1,6-bisphosphate. I know they like note the difference between the bisphosphate. To me just means that there's two phosphates on different different places, and di usually refers to them being on the same place. Is that always true? I'm not sure. I generally always thought of it like that, and it seemed to work out. So I think that's fair to use when you're thinking about these. But yeah, again, some of these reactions are pretty straightforward and just require memorizing, right? Like this one, for example, is also a very negative delta G step, one of the three controlled steps, which are 1, 3, and 10. This is step 3, right? And it makes sense that it's very very controlled because it's a very negative delta G value. Once you go past this point, you can't really go back, right? It doesn't really work. So you can't go back with the same enzyme, I should say. So having this ATP being used as a coupling agent with this fructose 6-phosphate to fructose 1,6-bisphosphate, it's probably not favorable to add another phosphate group to this to form a phosphoester, right? But using this ATP with a negative 30 kilojoule hydrolyzation value, right, makes it so that it's totally okay to do this reaction, right? We still end up with a very negative delta G value. PFK2 is also there. I'm not sure. I didn't see it down there. I'm sure it's in there somewhere. I just didn't see it. But if we see it, we'll move on to it and we'll get there. It's probably in regulation of glycolysis if I had to guess, which is the next one if I'm not mistaken. But anyways, yeah, hopefully this reaction makes sense. These reactions I would say are relatively straightforward. There's really not much to go over here on this. So we got aldolase and triose phosphate isomerase. So aldolase is doing a retroaldol reaction, and all that does is it takes this thing and it splits it in half, right? And you get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And then the role of triose phosphate isomerase is to turn dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Right? And glyceraldehyde 3-phosphate is the one that actually goes through the rest of glycolysis, basically. Right? So again, I don't think there's really much I can explain here. This is one of those things where it's just like you kind of have to memorize it. Of course, if you have questions, you can leave comments on it right on the video. I can more than happy to try and answer, or you can just find me in the IC if you have like specific questions about these reactions. But this lecture is very much a case of just like, here is the enzyme pathway. Have at it. I would say like this is one of those things where you just got to memorize it, right? There's another one in step 9 called enolase. Or I guess steps 8 to 9. I guess no. Step 9 is enolase. Right. So aldolase, triose phosphate isomerase, turning dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Right. That pretty much ends the investment phase of glycolysis. So now we're pretty much moving on to the payoff phase, right? And what you should actually keep in mind about this is we still have these intermediate, so we now have this glyceraldehyde 3-phosphate. We have 2 of them, right, that has this phosphate group on it that is now ready to be cleaved off in order to provide us with some energy. So that's kind of the main thing here. We're going to do a little bit more investing, but after that, we're pretty much in the clear. So they actually consider the next part to be part of the payoff phase, it looks like. So yeah, so actually pretty much done with the investment phase. So step 6 is interesting because step 6 makes this very high energy intermediate 1,3-bisphosphoglycerate. This is a pretty positive delta G value. And so you're probably thinking, well, man, why does it go to this product if it's a positive delta G value? Shouldn't it not go there? And to answer that question, we have to think about what we've been talking about for all of these reactions up here. Like, why are we phosphorylating? Why is it that's okay to phosphorylate this fructose? Why is it okay to phosphorylate that glucose, right? And the reason is because of coupling, right? So that reaction on its own is not favorable. But when coupled to another reaction that is favorable, then the overall reaction does become favorable, right? And so in this step here, it looks like it's not very favorable. We're adding inorganic phosphate. We're also oxidizing, right? And maybe oxidation is actually not too bad in terms of energy. It actually tends to lower it. But adding an inorganic phosphate, nuh-uh, not so much. That's definitely not very favorable. You usually don't just add an inorganic phosphate. Um, so that's a bit strange. So the question is, how do we do that, right? This is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. It is a dehydrogenase because it catalyzes an oxidation reaction. And the oxidation reaction I want you to keep in mind are from OChem, right? So if you remember chromium, if you remember PCC, right? Those things turn things into carboxylic acids and ketones respectively, right? This is a dehydrogenase enzyme using NAD+, which is also an oxidizing agent, right? And if you look here, right, where is the oxidation actually occurring? The oxidation is occurring here on this carbon. We are turning this aldehyde into a carboxylic acid and then forming a phosphoester, right, by adding this phosphate group, right? That is how you should be thinking about these reactions, generally speaking, right? You should try and think about them organically, right, in terms of the mechanism that they're using. But this is how it's doing it, right? So the NAD plus is used to oxidize this aldehyde, and then this phosphate is added to that carboxylic acid, that carboxylate group that is now newly formed. So now we've created this very high energy molecule, right, that has two phosphate groups attached to it that we can now use to generate ATP. We've done this on both glyceraldehydes, by the way. So both glyceraldehyde 3-phosphates have been turned into 1,3-bisphosphoglycerates. We got two of them, right? So we now move on to the next step. And like I said before, case of memorizing it, right? Yeah, here they show you the mechanism for this. I honestly wouldn't be too worried about this. I can maybe in like a problem set they ask you about this, but like other than that, they really should not be asking about this on your exam. There are better things to be testing you on. But hopefully this reaction makes sense. It's a bit complicated, but it's just like a general mech. You basically turn the cysteine to a nucleophile, right? And that attacks this gap to make this kind of thioester intermediate, or this thio intermediate, right? And then you basically form this oxidized intermediate, right, where you turn into 1,3-bisphosphoglycerate, right? So here, right, is now where we get to the coupled step. So I just said that going from this to this is not very favorable. The plus delta G, not favorable, right? But this reaction going from phosphoglycerate, 1,3-bisphosphoglycerate, to 3-phosphoglycerate is very favorable, right? Very negative in delta G, right? And if we couple these two reactions, basically we make them occur in quick succession, right? This overall reaction, if we add these two together, is quite favorable, right? So even though going from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate is not favorable, by coupling it to this formation of from 1,3-bisphosphoglycerate to 3-phosphoglycerate and forming ATP, right, we actually do generate a spontaneous reaction, right? And you may be wondering, well, man, how are we able to generate ATP? Well, we're able to do that because this reaction is so negative. It's so negative in comparison to the ATP, hydrolysis. I think there is actually a diagram, right? So if you look at this, right, ATP is at negative 30, right? But the 1,3-bisphosphoglycerate is at like negative 55, right? It is ridiculously negative how easy it is to turn this 1,3-bisphosphoglycerate into 3-phosphoglycerate, right? So it's very, very negative, right? Is the main idea, right? So it's totally okay to do that. So this step here is obviously catalyzed by glyceraldehyde 3-phosphate dehydrogenase, as we talked about, or GAPDH. And this second step here, the 1,3-bisphosphoglycerate to 3-phosphoglycerate, is catalyzed by phosphoglycerate kinase. And it's called a kinase because it is adding a phosphate to ADP to turn it into ATP. More often when we talk about kinases, it's taking a phosphate from ATP and adding it to something else. This is a bit of a reverse, right? This is the reverse because now we're going from 1,3-bisphosphoglycerate to 3-phosphoglycerate and putting a phosphate on ADP and not the other way around, right? So this is a bit of a weird one. You just got to wrap your head around it. Pyruvate kinase works in the same way, which is step 10, right? That's the main gist here, right? Because going from 1,3-bisphosphoglycerate to 3-phosphoglycerate is so much more negative than how positive it would be to turn ADP into ATP, right? It's so negative, right? To do that 1,3-bisphosphoglycerate to 3-phosphoglycerate reaction, it's totally okay, right? To form this this ADP into ATP, right? If you want to think about ADP to ATP as a plus 30 kilojoule reaction and the 1,3-bisphosphoglycerate to 3-phosphoglycerate as a minus 50 kilojoule reaction, the overall reaction delta G there is minus 20 kilojoules, right? Ridiculously negative still, right? So that is the main idea. And that is substrate level phosphorylation, right? Because we are using substrates, right? Substrate level phosphorylation is we're using other pieces to phosphorylate things, right? So ATP going to, like, I don't know, to like PFK1 would technically be an example of that. But in this case, right, we're using a substrate of this reaction, right, to phosphorylate ATP, right? That's the main thing you should get from this. And I'll get here, they actually show you the exact math for this too. So, you know, this reaction here, plus 6.7 kilojoules per mole, not very favorable, but this reaction, very favorable, right? Negative 18. Dang, very, very negative, right? If you add them together, right? Remember how to add these, right? You got to make sure that you're doing the canceling out method. So if we look at this, right? Let's see which ones we can cancel out. So this and this cancel, right? What else cancels here? Is that it? That actually looks like it's it. So that those two cancel, right? And what you're left with is GAP, PI, NAD plus on that side with ADP. So GAP, PI, NAD plus ADP. Wow. With NADH, ATP, and 3-phosphoglycerate. NADH, ATP, and 3-phosphoglycerate. If you add these two together, right, you get negative 12 kilojoules per mole. So, very favorable. Because they are coupled together. They are added together. If they were not coupled and they were separated, right, this would just be positive and that would be negative on its own, and they would not add. But then glycolysis would just not work, right? So, because the fact that they are coupled together means that this reaction actually occurs. Step 8 is phosphoglycerate mutase, right? Step 8 in glycolysis. And all this is doing, it is just taking the phosphate on 3-phosphoglycerate and moving it over here. It is a mutase reaction, right? And mutases are slightly different from isomerases because they only move one group, generally speaking. They don't really move a bunch of things at the same time. They just move one group, generally. We're going from 3-phosphoglycerate to phosphoglycerate. That's it. And funny enough, you guys have seen actually 2,3-BPG, right, as actually one of your things that actually affects hemoglobin, right, or O2 binding, and that is actually a glycolysis product, right? So 2,3-BPG comes from glycolysis. That's actually a byproduct of glycolysis, but basically it occurs in erythrocytes. And the way that this occurs is that once you get to 1,3-bisphosphoglycerate, you have a mutase, bisphosphoglycerate mutase, that turns this into 2,3-bisphosphoglycerate. And then if you want to go back to 3-phosphoglycerate, there is a 2,3-bisphosphoglycerate phosphatase and phosphatases, right? They just remove phosphate groups, right? So this one removes this phosphate group here, right? And it turns it back into it removes this phosphate group, it removes that phosphate group and turns it into 3-phosphoglycerate, right? So this is usually found when the pH of the blood is high. And would you really need to know this for an exam? Probably not. Might be helpful to keep in the back of your head just in case, but I personally have never seen them ask a question about blood pH with relation to 2,3-bisphosphoglycerate. Um, so and also, yeah, I do want to specify that this is usually found in the erythrocytes. I'm sure it can be found in other cells as well, but it's usually when we talk about the bisphosphoglycerate mutase, it's usually met in erythrocytes and not other cells. Um, and this is the kind of like the idea of respiratory alkalosis, right? So you guys talked about this before with hemoglobin, um, but basically, right, if you exhale a lot, right, you drop your CO2 levels, which drops this, which all shifts this way, so H plus drops, so you get more basic solution, right? It's all from the first exam. And the Bohr effect, right? And again, I feel like you guys are probably not going to be tested on this stuff again until the final. But generally speaking, right, this is, if you remember how the Bohr effect works, I'm not going to go over that in depth. I already talked about it in previous lecture videos, but just as how the pH has some varying effects on hemoglobin and those O2 binding proteins. Back here, right, got your T state and your R state, right? T state, R state, and then if you were to look at the what does base do, what does acid do, how does it change, right? Right. And we're back here again, and again, so like there's a lot of slides in here where I don't feel like are that important, to be honest with you. I think the main one, the basic thing you should take from this lecture is you need to memorize glycolysis. Those 10 steps need to be memorized, and that's like the main thing that you guys should be doing for sure. But yeah. Now we move on to enolase and glycolysis. So basically what you should know about this is, I want to double check the reaction type that enolase does, because it is a dehydration reaction, I believe. Yeah, it is. Yeah. Basically, right, it is a dehydration reaction. There's like there's a funky little mechanism for it that I can't remember off the top of my head, but trying to remember. It's one of those reactions you learn in OChem base. But what we should know from this, right, is that enolase, right, basically acts as a basically acts to turn this into a kind of a reactive intermediate, right? Which is phosphoenolpyruvate, and then phosphoenolpyruvate is turned into pyruvate, which is a negative delta G enough drop to actually form ATP from ADP, right? So what's going on here is we have our 2-phosphoglycerate. We're removing this H and we're removing this OH. That turns into water, and you form this new double bond here, right? So now we've got this phosphate, and now if you look at this group here, this is actually an enol, right? And if you remember from OChem, enols can tautomerize into ketones, and ketones are very, very stable, right? And the delta G for forming a ketone from an enol is pretty dang negative, which is why we're going from phosphoenolpyruvate to pyruvate is so favorable, because this is a really nice reaction to do for that keto-enol tautomerization, right? And that drop in energy is enough to drive the formation of ATP from ADP, right? So that's the main thing I want you to get across from pyruvate kinase. So enolase, right, is responsible for making this kind of reactive intermediate, right? It's still a negative delta G drop from 2-phosphoglycerate, right? But it's enough to make this it's a small delta G, negative delta G drop from 2-phosphoglycerate to phosphoenolpyruvate, right? And this phosphoenolpyruvate to pyruvate is an extremely negative delta G drop, right? This is step 10. This is one of the other regulated steps in glycolysis, right? It's very hard to go back the other way, practically impossible, right? But it's so negative because of that keto-enol tautomerization that occurs. But yeah. And here, right, we've got an example of how this works, right? So if you see here, pyruvate kinase, right, you use this phosphate to grab this ATP. And basically what's going to happen here now is that the electrons, once this phosphate is gone, this is going to go down here. This is going to grab an H from the solution, right? Pretty much is how this works, right? You end up with this product here. And yep, so we've got from phosphoenolpyruvate to the enol form of pyruvate, right? It's a pretty negative delta G drop, right? And from there to pyruvate, it is insanely negative. It's like negative 50, right? So this is an overall like a negative 60 something kilojoule reaction. So it's a very negative reaction, right? And that is the fun part of this glycolysis. And that's actually pretty much the end of glycolysis. There's some other things that come afterwards, right? But for that, make sure you memorize all of those reactions, right? But now we're going to move on into the other parts of this, which is talking about this. Yeah. So this balanced equation, you actually should be able to derive it as you go through glycolysis. So the way you know that you've mastered glycolysis is if you're able to, one, list out all of the reactants and products for every single step in glycolysis, and two, if you were to look at an overarching idea of what glycolysis produces, uses and produces, right, you should be able to make this equation, right? You don't have to know this value exactly, but generally speaking, you should know, this equation, what goes in and what comes out. And like, we know what steps are all of these happening in. So the ATP consuming steps, yes, we're going step 1 and step 3, right? Hexokinase and PFK1, right? ATP yielding steps, right? So going from 1,3-bisphosphoglycerate to 3-phosphoglycerate, and going from phosphoenolpyruvate to pyruvate, right? Those are all step 6 and 7, and then step 9, right? Those are ATP yielding steps, right? And we've got some phosphorylasis reactions, isomerizations, right? So where the isomerizations, phosphoglycerate, you got triose phosphate isomerase, right? Phosphoglucose isomerase, right? Where are the oxidations occurring? The oxidations are occurring with GAPDH, right? Glyceraldehyde 3-phosphate dehydrogenase. The lysis reactions, enolase and aldolase, right? And the dehydration reactions are occurring with enolase, right? So you should be able to pinpoint exactly where these reactions are occurring and what their products are, right? And that should be what you focus on when you try and learn glycolysis. So now we get to the question of, okay, we've turned this NAD plus into NADH. Don't we just run out of NADH eventually? Well, no. That would be weird, wouldn't it? Because then you'd be dead. You wouldn't have anything to use. So there are ways you can actually regenerate this NAD plus for that step 6 reaction, right? The glyceraldehyde 3-phosphate dehydrogenase reaction, right? And those ways involve things like, the lactate pathway or the ethanol pathway. And then you also go down into citric acid cycle that produces NADH and FADH2, right? So there are ways of regenerating this NADH, right? So let's start with lactic acid fermentation. Very simply speaking, when you don't have any oxygen, right, your cells have to still do work. It's also why you feel like fatigue and you feel like a burning sensation is because you have a bunch of lactate in your body, right? And lactate or lactic acid, lactate is actually the basic form, right? Is a deprotonated, like a very acidic a relatively acidic compound, and once you make lactate, it actually puts a bunch of H plus in your blood, and then you get this feeling of dizziness, and you want to throw up and all that jazz after like a run or something, right? So, if you have no oxygen and you can't do citric acid cycle and electron transport chain, the glucose, after being turned into pyruvate, is reduced into lactate, and there you go. There's a reaction, right? And when it is reduced into lactate by the NADH you made in step 6, and it is turned back into NAD plus to allow it to go through that cycle again for step 6. So basically what's going on here is glucose into pyruvate into lactate, uses that 2-NADH, right, turns it into NAD plus, that 2-NAD plus is used again in step 6 to turn glucose into pyruvate, and you basically keep doing that over and over again until you run out of glucose, right? Here is that reaction, and here's a nice structural image of how this actually works. This is just a reduction reaction, right? This H comes in, attacks this comes up, right? And voila, you're left with this. I know the OH is flipped around. It does not actually matter, but you make this, right? And this enzyme is lactate dehydrogenase. It uses a oxidizing / reducing agent, right? In this case, it's a reducing agent, right? So it is a dehydrogenase. It's an oxidoreductase reaction, right? And very negative delta G reaction, right? Very favorable to do this, right? So something else to keep in mind. And remember what I said, right? This is under conditions where glycolysis is the only source of ATP, right? There's in the muscle cells, hypoxic conditions. In erythrocytes, actually, a very important fact for the MCAT, they don't have mitochondria, so they only do glycolysis, right? And there's some bacteria that do lactic acid stuff, and then some tumors as well only use glycolysis, right? So this is something to keep in mind, right? This is just a reduction reaction, right? It's a reduction reaction in the sense that we use the NADH as a reducing agent to regenerate this NAD plus to use it again as an oxidizing agent in step 6 of glycolysis, right? Ethanol fermentation. So if you've ever had alcohol before, right, this is how you make ethanol, right? So ethanol, right, yeast, right, is another way of producing this NAD plus again, but this is usually specific to yeast, right? Basically, right, the first thing that happens, right, is you end up with you have a decarboxylation reaction. And this is actually a nice little sneak peek into any future reactions you do. I believe with, I believe pyruvate dehydrogenase uses this cofactor TPP as well. I have to review that a little bit before you guys cover it. I can't remember which lecture that is, but basically, right, TPP, right, if you ever see a decarboxylation reaction, think TPP. Similarly, if you ever see a carboxylation reaction, think biotin. And you're probably looking at me like, what the heck are you talking about right now, Min? But trust me, it'll come in handy eventually, right? TPP, think decarboxylation. Biotin, think carboxylation, right? And what do I mean by decarboxylation and carboxylation? Carboxylation, if you use biotin, is just adding a CO2 to something. Decarboxylation in this case is the removal of CO2, right? So if you look at this, TPP, right? It is a decarboxylation reaction, so CO2 is leaving, right? And so now you have this, you form this acetaldehyde, right? Because you're getting rid of a carbon, right? If you want to think about how this mechanism works, you can think of it like this, this goes off, goes here, right? And then that electron pair goes up and grabs H from somewhere. That's one way of thinking about it. Is that how it works? Probably not, but it's one way of thinking about that reaction if you want to think of it in terms of OChem, right? So that's pyruvate decarboxylase, right? We're using magnesium here, right? Because of a negative charge up here, right? And also TPP itself, I believe, is a negatively charged cofactor. Well, generally speaking, these decarboxylation reactions tend to use magnesium, if I'm not mistaken, as well as TPP. So we form acetaldehyde, right? So we haven't regenerated our NAD plus yet, but here's where we get our NAD plus generated. From glycolysis, we use this NADH to reduce this carbonyl into an alcohol, right? So if this attacks here, these come up, right? And you're left with this, which is ethanol, and your NAD plus has been reformed, right? Because you've just reduced this using NADH. Now you have your NAD plus again, and you can go ahead and do that glycolysis pathway all over again. And the only way this changes the reaction is by adding ethanol here instead of pyruvate, and then adding some CO2 here as well. Right? The rest of it actually pretty much stays the same. And here is actually TPP. So remember how I said magnesium, right? Notice how there's a double phosphate. diamine pyrophosphate, right? So double phosphate, the magnesium, right, is actually very important for forming these coordination complexes with these phosphate groups to stabilize it, right? That is why we have a phosphate there. So, do you have to know this reaction? No. Highly doubt it. If your professor said to know it, that's crazy, but I guess sure, but you should not need to know this reaction for the class. Right? Yeah. Several. Yeah. We were studying decarboxylations, right? That's what I said. So TPP is for decarboxylations, generally speaking, right? And there are many TPP dependent reactions. So pyruvate dehydrogenase uses it, like I said, right? Because we have to turn this into acetyl-CoA, and notice how we lost this CO2 group. Pyruvate decarboxylase, we've lost this CO2 group. Transketolase, right? This is a bit of a different pathway, right? But it's along similar lines. In the pentose phosphate pathway, which you'll be covering a little bit later. But that's actually the end of this lecture. So you should be able to remember these, right? Please, please, please spend the time to memorize these reactions, right? You will need them for the exam, and it'll save you a lot of points. Especially if you've looked at my 2024 exam, the one I took when I took this class. That exam is very heavily dependent on whether or not you actually understand the pathways. Very heavily dependent on whether or not you understand the pathways. I will be doing a workshop on that exam on Monday, so tomorrow, the time of recording this, right? But I want you, hopefully you have these reactions memorized by then, right? Because you need to have them memorized because your exam's on Wednesday, and the sooner you have them memorized, the better, right? So, yeah. What types of enzymes catalyze them? So is it a mutase? Is it a kinase? Is it a dehydrogenase? Is it a cleavage enzyme? Is it a lysis enzyme? I don't know, like enolase, aldolase. What kind of enzyme is it? And what additional B vitamin? You should actually know the vitamin name. So TPP, like thiamine, right, is actually vitamin B1. So TPP is B1. I don't know if you need to know the exactly like which B vitamin it is, but it does say B vitamin, right? So just think that TPP is B1. Biotin, when you start learning about biotin, is actually B7. So yeah, and then how do those enzymes work in fermentation? And why are there different types of glucose transporters, right? Why is GLUT1 different from GLUT2, which is different from GLUT3, which is different from GLUT4, right? GLUT4, right, for muscle, right, high affinity, needs that glucose fast. GLUT2, for the liver, they don't need to store every bit of glucose, right? So they have lower affinity, right? And GLUT3 for the brain needs 40% of your ATP stores, right? Need to use a lot of it. And then GLUT1 is just general purpose, right? So being able to describe why these things occur and why you need them is a very important skill to have, and it could easily show up on your exam. Hopefully I didn't talk too fast. I feel like I was yapping quite fast. I wanted to make sure I got this video done in a decent amount of time. This is a long one though. Hopefully it made sense. And thank you guys for watching. If you have any questions, feel free to stop by the IC or to post a comment on the video. And yeah, happy studying.